I switched gears a little bit, and I'll try and give you a little bit more of an overview of how we got into this sort of question of figuring out how microbial genomes are really structured in the environment, I would say. So bear with me, and we'll eventually end up with the viruses as promised. Okay, so first of all, let me set the stage a little bit. Many of you are, of course, well aware that there's a tremendous amount of microbes out in the environment, but maybe not the full scale, right? And so there's really, globally speaking, I would say this planet is very much a microbial one. And this is really, I think, very well illustrated if you look at numbers of microbes in different environments. And so you will see here that in the ocean and in fresh water, there's about 500,000 to 1 million microbial cells per milliliter of water. Okay, so I always tell students or people, if they worry about bacteria in the environment, this is the best sign that almost no bacteria in the environment are pathogens, because you would be dead if you go swimming otherwise, right? Because you ingest bacteria all the time. So if you scale this up, there's an incredible amount of microbial cells in the marine habitat alone. But then the biggest actually reservoir of microbial cells, by far the biggest one, is the deep subsurface. So bacteria can be detected down to 3,000 meters depth. And we think that really temperature actually limits microbial life, and perhaps pressure as well. And then there's animal guts. And even though we can be packed full of microbes, globally speaking, it's actually fairly small. And so if you think about the living biomass that's present in microbes, it's actually quite similar to the living biomass in plants. So not if you count the wood and so on, but the protoplasmic biomass and so on. And so these microbes, why do we care about them? We care about them because they actually carry out numerous environmental functions that enable life on the planet. They've been here for a long, long time. So they act as, what I view the main function is really that they act as chemical catalysts. And they catalyze a tremendous number of different kinds of reactions. As you can see here, a small sampling of those. And we care because we really need to understand, I think, how communities actually achieve these reliable ecosystem services that we call them sometimes. And then what are the limits, especially in face of environmental pollution, in face of global change? It becomes more and more important to understand when this system actually comes out of whack and when things break down. And so this creates this need to really understand microbial system composition and dynamics. And so in a way, the classical ecological approach is that you go out into the environment and you figure out what species are there and how they perhaps interact with one another. So if you have, let's say, one population of one species going down, how does this permeate through the system, and what are the consequences for the ecosystem functioning? And so in a way, this network of species, these interactions, that resembles a bit, I'm always reminded of gene interaction networks. But the only problem is that in microbes, we don't quite know what the unit of the interaction should be. In fact, especially if you're interested in functions. Is it a gene, or is it populations? Is it species? And so really the fundamental problem that we face in microbial ecology, or one of the fundamental problems, is that we don't have a good idea of what a species is. So what's this unit? A species or a population, the locally coexisting members of a species? We don't understand this very well at all. And so the main reason for that is that gene flow, in principle at least, knows no bounds in microbes. So you can have, in principle, any microbe on the planet can exchange a gene with any other microbe. There's no strict gene flow boundaries, at least in principle, I will say. And I'll show you later an example that we do think that there's some relevant boundaries actually that allow us to classify things into species-like units. Okay, so DNA uptake is can take the shape of different kinds of mechanisms. It can be by transformation. That's the uptake of naked DNA from the environment. Transduction, DNA transferred by viruses is very, very important, especially since viruses can take up also genome parts from organisms and transfer them. And then conjugation by mobile genetic elements like plasmids can transfer DNA between cells. Once the DNA is inside the cell, it can actually, most often it's actually destroyed, I should say. And I'll speak a little bit to that in the latter part of the talk. But if it is taken up into the genome, it's by two principal mechanisms. The first one is homologous recombination. In this audience, I of course don't have to talk much about this, but very relevant is that for homologous recombination to act at high rates or with high efficiency, the sequences need to be quite similar. Okay? So this is already one limitation of this limitless gene transfer potentially that we see in the environment. And then there's integrase mediated transfer or illegitimate recombination that has short recognition sequences or no recognition sequences at all, and that can act on dissimilar sequences. And so the effect of this gene transfer, we actually see quite a lot when we look at bacterial genomes and at gene phylogenies, for example. So one of the things that people have seen over the years is that when you look at different genes across the genome, so these gene trees for a group of bacteria, and you see that depending on the gene that you actually look at, you will get a different phylogeny, and sometimes it groups totally different strains together. So phylogenetic trees can be actually unreliable if you're interested in sort of the descendants amongst microbes, and especially the further back you go into time. And the second part is that large genome content differences among otherwise very closely related genomes can arise. And this is a really classic example of 3 E. coli genomes that were sequenced now a number of years ago. But I always like to this slide to illustrate this point because it's a very general one amongst microbes. These 3 genomes that are all E. coli only share 40% of their genes. And the other genes are only present in one of the strains or in a couple of the strains. And this relationship we call the core genome, that's the shared genes. And then the flexible genome are the strain-specific ones. And then the sum of these flexible genes is the pan-genome. And it's thought to be very important in rapid adaptation of microbes to environmental factors. So this problem of not being able to categorize neatly bacteria into species has led to some quite arbitrary classifications amongst microbes. And you will see this in the microbiome literature all the time. The use of 16S ribosomal RNA, for example, to classify bacteria. And people will say these represent species, but that's certainly not true because this is an extremely conserved gene. The thought is that you accumulate mutations at a rate of 0.5% SNPs will accumulate in the space of 1 million years or so. Okay, so you can imagine that once you accumulate mutations in the 16S ribosomal RNA, you're pretty far back in time already. And so it is thought that you at best, can classify the genus level with the 16S ribosomal RNA. In bacterial epidemiology, however, much higher resolution approaches were actually taken before genome sequencing was so cheap. Multi-locus approaches were taken, and now people mostly actually sequence genomes right away. And so what my lab did over the years was we tried to transpose these sort of approaches from epidemiology into environmental questions and into the environmental context. And I'll show you an example of that, how we got at a fairly good prediction of what we call environmentally differentiated populations some years ago already, way before genome sequencing was cheap. So the first question I want to address then is, can we actually identify ecologically cohesive populations that we can then use to learn more about the properties that link them to environmental factors. And so I'm showing this is a quite old example, but I want to show you also how a relatively simple approach taking sampling into account can be very, very informative, even if we do not, even if we have to be completely agnostic towards species and also really the true habitat of the organisms that they occupy. So the complexity of the problem is illustrated a little bit in this slide. When you think about the marine environment or an aquatic environment, you may think that it's quite homogeneous, but that's actually not the case. Okay, so when I take a bucket full of water, it really contains really a universe of ecological opportunities for bacteria. So first of all, you can swim around as a free living cell and live on dissolved organic material, basically. You can attach to different kinds of organic particles that are floating around. There's, of course, organisms poop in the water, they die. There's all sorts of particles and slimes that are floating around. Don't want to like scare you, but ocean water is literally full of that stuff. And so are lakes. Bacteria attached to them degrade them. They're attached and associated with different other kinds of organisms. And they can, of course, migrate between those. Okay? But there's definitely the potential for specific associations. And so when we got into this, we thought it's nearly impossible to pick individual organisms and particles out of the water column and trying to figure out what is associated with them specifically. And so we took a pretty simple approach where we just size fractionated the water. And the idea here was that you could sort of operationally separate or enrich different kinds of associations here. You could have a size fraction that is most clearly free living, one that might be a little bit ambiguous, containing larger cells or cells attached to smaller particles, then things attached to larger particles or smaller organisms, and then larger organisms. And then we used culturing on selective media to isolate vibrios. That's a group of marine bacteria where we just had very good selective media for. So when you throw ocean water on there, these bacteria on average will actually grow on nothing else. So that simplified the problem of picking all sorts of very diverse organisms. So we did this. This was completed in 2008. And we picked about 1,000 isolates and sequenced protein coding genes. Remember sort of this rationale to get a higher resolution than the 16S, constructed phylogenies, and then mapped the categories, the size fractions from which they were isolated from onto the tree. And you see already that these categories are not homogeneous across here. So we did this also in 2 seasons, I should say. So here you have the different seasons, and here you have the different size fractions. But in many cases, it's not really clear if there's a boundary or if this to be drawn that denotes a different ecology. And so we developed a model to support this. And so don't read this. This is a little bit too much text here. It's an old slide, but it's a fairly simple model in its assumption. So it assumes that bacteria have sort of a true habitat, but it's an unknowable category because of the complexity of the environment. Okay, and so then you can any node in the phylogeny can denote a transition, basically, where evolutionarily a population switched a habitat association. Then the second assumption is simply that our sampling scheme leads to a characteristic distribution of the strains that reflects an underlying distribution of the habitats. So let's say, for example, this population is mostly free living and attaches a little bit to some particles. You will see this in the sampling scheme here, okay, and so on. And then we have the phylogeny, and then you can basically the model then decides basically when you switch from one characteristic distribution to another one. So very simple, basically very simple sampling scheme, phylogeny, combine the two, and see where you get to actually. And so what this actually led to was then a very clear hypothesis of different habitats in a sense. So here you have these guys are free living. And then interestingly, we found that even when there was very little genetic differentiation, you could have very rapid habitat switches. I don't have time to go into the details, but we spent a good number of years trying to then figure out if this is actually true. And in most cases, this has actually held up remarkably well. So this underlying distribution, taking this into account, really led to a very solid information about population structure. I should also say that like this entire branch here represents perhaps a 16S ribosomal RNA. So 16S ribosomal RNA cannot give you really the relevant ecological information. The strongest clue, though, that this actually was giving us some really reflecting some ecological processes, evolutionary processes in the environment, came from the finding that we were able to reconstruct some speciation and adaptation events from this sampling. And I want to show you this example here. We had a couple of clades where we had almost no differentiation, and the two size fractions were clearly separated from each other. So this is the largest size fraction, and this is the one of the lower ones. So there's size fractions in between. So we thought it wasn't like blending or bleeding over, where we had sort of fuzzy boundaries. This was also quite some number of years ago, but it was already the kind of early days of next-gen sequencing. It was in 2010 or so when we began this. And we picked one of these differentiations here and then picked strains and sequenced their genomes. And the interesting thing was when you do whole genome phylogenies, you recapitulate actually the ecological split very well. So we thought, oh, that's great, you know, we're done. But Jesse Shapiro, who spearheaded this, he then actually looked at the recombination dynamics across the genomes, and it actually became a lot more complicated. I should also mention that these are really super closely related. So they're 99% identical across the genome in nucleotide sequences, but yet they retain this sort of property of very large flexible genomes. Okay, so there's these private genes that each strain actually has. The next slide is probably the most complicated one in this whole talk, but bear with me. Well, actually, probably not for you. You're so well versed in genomics, of course. So here is the number of SNPs that support the ecological split in the tree that I showed you previously. And here's the chromosome length. The Vibrios have 2 chromosomes, so this is the smaller chromosome and this is the bigger chromosome. And one thing that Jesse noticed right away was that when you look across the genome, there's almost no support for the ecological split. And in fact, when you look, he then developed a method to look at recombination blocks. So blocks in the genome that have their own phylogeny essentially because they're recombined. You see that every one has its own phylogeny and it doesn't split up the green and the red at all. And each one of them, this is cut off a little bit, I apologize, really accounted only for a very small fraction of the total number of trees. Okay, but then we saw that there were these regions in the genome where the support for the ecological split really came from. So where these populations, as I call them now, were very clearly differentiated. And you see the tree structure here. What we found was that in at least one of the populations, the strains were nearly identical, and here in both populations. Okay, and so the way we interpreted this was that what happened here was that a genome region was acquired by horizontal gene transfer from a divergent source, because also look at how long the branches are here. and then swept in a population-specific manner. Okay. And so when you look at annotations in the genome, we saw that they did actually contain genes of potential ecological relevance, and most importantly, for biofilm formation. And we were able to confirm this later on. The red population actually really likes to attach, whereas the green population is highly modal and keeps swimming from resource to resource and kind of never stops. Okay, so that's really one of the driving forces behind this differentiation that we saw, and it's consistent with the sampling that we saw, because the red population, red, remember, denotes large particles or organisms, whereas the green population is free living or small particle associated. So how is this possible that genes sweep through a population when they seem rather mixed across the genome in terms of the gene phylogenies and the recombination? And so we were sort of lucky that we had, in each population, we had pairs of strains that were very, very closely related. And so the thought was that these differentiated after this ecological split actually happened between the populations. And so what this enabled us to do was then to look at the number of recombinations that these pairs had with their own color group and with the other color group. And in each case, what we saw was that the recombinations from within the population were actually enriched. Okay, and so the way we interpreted that was that that the most recent recombination, so that this mixture of phylogenies that we saw previously was sort of the history of recombination. But when you look at the most recent recombinations, that they have already become population specific. And so we came up with this model then that thought was a fairly decent explanation for this process, that you can sort of see a three-step process. The first one is that some of the genomes receive some adaptive mutation. The ones that we could identify all originated by horizontal gene transfer, I should say. We've seen this over and over again. The bacteria actually really adapt by horizontal gene transfer. They just, there's so much material out there that can take it up, and somebody else has probably tried this function and optimized it already, and you just pop it in a modular way into your genome and use it. In this process of this very, very sort of beginning adaptation, you can't really resolve in the genomes yet, because there's no signal. But then, of course, if this leads to an ecological differentiation amongst the two, or to some other sort of trade-off, okay, then you can imagine that you can sort of get a splitting of the gene pools. And the ecological split is probably the first step to decrease the recombination rates. And that can be then reinforced by mutations that accumulate in a population-specific manner. And so here you might get some, in a phylogeny, some sorting according to habitats, and then this is when the speciation really has kicked in, and you get some sort of different trajectories of the population. And so I bring this example, first of all, to show you, how we're trying to figure out how to go from genomics now to really hypothesizing also ecological adaptations, but also because I'm going to come back to this idea that horizontal gene transfer and then this population-specific sweeps that we see in genes helps organisms adapt very rapidly to also viruses. Okay, so I'm going to switch to viruses, and I'm going to give you also a little bit of an idea of the planetary proportions here. So these are the number of viruses we think that are present in the ocean, 10 to the 30. And I want to put this in a little bit of context for you. We're going to scale this by their average length, which is about 100 nanometers. And that gives you that the viruses, if you line them up in a single line, will give essentially a length of 10 to the 20 kilometers. So now think about that. That's 10 to the 7 light years of viruses. And our own, so the nearest star is about 4.2 light years. And our own galaxy is 1.5 times 10 to the 4 light years across. So that's the number of viruses that are present on the planet. That's pretty incredible, isn't it? So they're the most abundant entities that are floating around everywhere, essentially. And they have a tremendous influence, particularly on microbes, because most of those viruses are, of course, microbial viruses. Oh, I popped in this slide when I was waiting for the talk, so I forgot that I had put another slide in there that summarizes the previous, but I don't want to break the flow now. So viruses are incredibly important in the marine environment. They're abundant. There's about 10 to the 7 viral particles per mil. I mentioned that there's about 1 million bacterial cells, or 500,000. So there's about 10 times more viruses than bacteria. And they're thought to really alter carbon and nitrogen cycling because they attack these chemical catalysts, bacteria, and they generate dissolved and particulate organic matter that can also sink to the bottom of the ocean. And once organic matter sinks to the bottom of the ocean, it remains there for quite a long time, actually. So it's kind of a very important climate regulator in that sense. But there's this problem that viral predation is thought to be actually very highly strain specific, and bacteria are highly diverse. So there's really, it's not very well understood, even though people will say that viruses are very important in regulating the ocean system. How this actually works is much less clear. Okay, so how does this killing all work? I mean, viruses are supposed to, consume about 20–30% of bacterial production each day. So counting doesn't tell us, because we can't really link viruses to the host. Sequencing doesn't help much either, because most viruses are completely unknown, and even if you know them, you don't know necessarily what their hosts are. And so what we decided then was that we need live bacteria and viruses to try and really understand how at least the specificity and interaction works out. So a former student in the lab, Catherine Kaufman, she decided to do an incredible experiment. She went out into the coastal ocean. So this is the Boston region, and this is Nahant. This is a small peninsula that juts out. And so by wading into the water, you can pretty much sample ocean water. That's not very influenced by fresh water input from land and so on. And she went there from our campus at MIT. It's about an hour drive every single morning for 93 days and sampled water every single day. I always said, Catherine, take a day off. And she always said, if I do, I will never start up again. And she sampled water, she isolated bacteria, she archived viruses and microbial cells. And so we have this incredible time series now where we have bacterial isolates, viruses that were co-isolated, and then a cross-infection matrix that was conducted. And it's the largest virus bacteria cross-infection matrix there is, and it's fully genome sequenced. So we have about about 300 phage and 1,000 bacterial isolates sequenced from that time series. Because she did the isolations in a quantitative manner, so she basically precipitated out and flocculated the viruses, created a concentrate, and then plated the viruses on bacterial lawns. And so the number of these are called plaques. That's when the viruses lyse a cell, tell you then also the concentration of the viruses in the environment. And so the first question then was what's the specificity of the interaction and what's the diversity of the viruses that we see. And so it turns out that collection alone, this was all done again on Vibrios, on our model system. So not a super diverse group of organisms, but we pulled out a lot of different viruses that represent, you know, I don't know how to define even a genus in viruses, but people do it. So the genus level diversity was incredibly high. And with many different representatives of the major groups. One thing that we noticed, though, when we look at the cross-infection matrix that these viruses produced, and what I'm showing here is the bacterial host, categorized by some protein marker genes, and then the different viruses on the y-axis here, that this matrix is incredibly sparse. So only these black dots here represent an infection, and when you see color, there was no interaction that we could detect between the viruses. And so there's a couple of broad host range viruses that we pulled out. This we described in a separate paper. And then there's what we call population-specific viruses. And strain-specific viruses where you only find an occasional hit. But even the broad host range viruses are very strain-specific. So when you look at closely related potential hosts, they're very specific to one of those oftentimes. So we were then interested in what determines the phage specificity. Why are they, in fact, so specific? And so in the past, people have focused very much on receptor variation. So viruses are, of course, non-living particles when they're floating around, that they need to inject their DNA or RNA, of course, into host cells. And there's these proteins and carbohydrates on the bacterial cell surface that the phages need to attach to and recognize, and then inject their DNA. And when you throw a phage or bacterial virus and a bacterium together in culture, you will immediately select for mutations in these receptors. And so the field was actually focused on these receptor mutations for many, many years. But I'll show you that this is actually not the main mechanism. And the clue to that was actually already there when we started this work from really findings that there's an enormous defense arsenal inside bacterial cells that has the potential to inactivate viruses. There's, of course, CRISPR-Cas that you all know very well, and restriction modification systems. But the last few years have seen really an explosion of discovery of new phage defense systems, abortive infection systems, where the cell actually commits suicide when it's infected, new defense systems that we don't actually even know the mechanism of action yet, and phage-inducible chromosomal islands, and so on and so on. And it's also been found that these defenses actually are quite variable from genome to genome. And this has given rise to this idea of this pan-immune system of bacteria, that there's these antiviral defenses that are really spotty in distribution. So one strain may have one, but not others, and so on. But the mechanism of how they get around and how it's actually structured was actually not very clear. And so we started out by asking this simple question: what determines the specificity? Is it receptor modification versus defense genes in the wild? And we basically reasoned that these dynamics are basically not really understood. So I want to take you back to this cross-infection matrix for a second. One thing that we noticed was that we had strains oftentimes that looked very clonal, so had no SNPs in these marker genes, yet they were infected by different viruses. And we had multiple occurrences of that across this matrix. And we decided to focus on one of those, this isolate collection here. We sequenced the genomes, and we found that in some, they only were differentiated by 17 SNPs. So they're really, really closely related, yet they were infected by two different viruses that are really completely unrelated to each other. Okay? And so the first thing we wanted to understand was how potential receptor modification or differences might actually influence these dynamics. And so what we did was we mapped the receptor. Too many animations, sorry. we did transposon mutagenesis as well as resequencing of spontaneously resistant isolates. And we found that in all cases, this mapped to a couple of genes. So for the one gene, this orange phage attaches to a general secretion pathway gene. And this other phage has two receptors. That's very common. So the lipopolysaccharide, that's this carbohydrate decoration that many bacteria have on their surface. And then it has a secondary receptor, protein that spans the membrane through which it actually then injects the DNA. Interestingly, that this is sort of a model of how this actually happens. This is the type 2 secretion system, membrane tunnel, and then here the first receptor and the secondary receptor. When we looked at the genomes of our strains, we saw that there were no SNPs in these, meaning that the receptors are invariant across the entire strain collection, and that the phages or the viruses always can inject their DNA into all the strains, which means then that the DNA of the phages must be specifically inactivated in the different strain groups. And so when we went back to looking at the strain phylogeny, so here's, of course, that's the core phylogeny. But when you look at the flexible genome, what you will see is that you get, when you do a presence-absence matrix of genes amongst these strains, you perfectly recapitulate the differential infection by the phage. And so we looked into the genomes and we found that this clustering according to infection is actually due to large mobile genetic elements that are specific to the two strain groups. I will spend a little bit of time talking about these because they're very interesting. I think they have very interesting biology that we don't understand at all. First of all, most genes, we have no idea what the function is. They don't annotate very well at all. And so the ones that do are in many cases were known phage defense genes. So we have restriction modification, toxin antitoxin systems, here again, restriction modification, and several other genes that aren't, whose annotation isn't shown here. They also have this interesting property. They have integrases, so they is an enzymatic process. They have recognition sequences, and they oftentimes cut a host gene apart when they insert. But when they do this, they actually duplicate that gene in their own genome. So they do not disrupt host function, which I find very interesting. So these are not really, this, like I heard a lot about transposons today. They're not really this sort of classical parasitic elements. I think they're well adapted to not disrupting the host too much. We then were interested in mapping the function of these, because why do they have several ones? And I won't go into so much detail here, but we knocked out in the orange strain group all three of these defense mobile genetic elements, and then retested them the susceptibility against the phage. And so this is a dilution series of a phage, and the strength of interaction can you can glean from when at which dilution you still get this clearing of the strains. So this is the phage against the orange wild type, and you see that you get only at the highest dilutions you get some clearing. And this is actually not due to reproduction of the phage. This is a common phenomenon. When you overload a microbial cell with many phages, you will kill the bacterium because you poke so many holes into the membrane probably, or because some abortive infection kicks in. When you try and isolate a phage from these clearings, so this is when you maybe stick a toothpick in here and try to restreak a phage, you will get no propagation, no viable phage. Whereas down here, in contrary, you see that you get clearing from that. So we knocked out, this was quite a lot of work, the individual defense elements in all possible combinations. And lo and behold, what we saw was that what you needed to really knock out all three to get the same level of susceptibility as the purple wild type towards that phage. So they really collaborate in defending this host. And the first level of defense, we think, is these restriction modification systems, but they're quite imperfect defenses. They're quite leaky. And so when the phage gets through, these other systems kick in and they actually kill the host. So they're kind of really an emergency brake. Okay. But the population, of course, is still protected because you don't get any viable phage out of this. Now, I mentioned that these are mobile genetic elements, and we were interested in what the dynamics are. We already saw that, you know, we have no SNPs across the genome, and you can have different components. So that means that the evolutionary turnover of these elements must be actually quite high. But we had water samples from the time series, and we developed a specific digital PCR assay where we quantified 3 of the elements in a way that spans the element and the bacterial genome. So you measure the sort of the clonal frame as well as the element. And what we saw was that in a mere time span of 60 days, there was a shift in the abundance of these different elements. So here we have an element that's present in the purple strains and one that's in the orange strains, and they're perfectly correlated. So there's no ecological difference in these strain groups. They go up and down together. But in the orange population, we saw that after about 60 days, there was a spike in this particular genome. We don't know if that's you know, de novo acquisition or a strain that already had it that became more prevalent, but it certainly is really indicates very, very short-term evolution in the environment. Like 60 days, you know, is really quite incredible, I think. The other thing we did was we expanded sort of the search for these elements, and we saw that these individual bacteria are full of different kinds of defense elements, as we call them. And an individual can have as many as 18 of these PDEs. PDE means phage defense element. That's an incredible burden for the host genome. And we saw that at least amongst closely related strains, about 90% of the flexible genome is constituted by these phage defense elements. So defending yourself against phage is really important. They fill up their genomes, and of course, you know, there's always a cost of autoimmunity when you carry defense genes, so there must be a cost as well. Okay, so I'm getting closer to the end now. I just want to give you an impression. We looked into other bacterial groups as well. This is just some pathogens. We sent the paper to Science. You know, reviewers always, we're used to this. They say, Vibrios, is this important in other bacteria? So we went and did it. And sure enough, they have also these defense elements, and they carry defense genes, and they vary just as quickly. So this, we think, is a very common phenomenon, how bacteria defend themselves. Now I'm coming back to these gene-specific sweeps, and this sort of notion that receptors are important, or receptor variation. We identified these receptors, and then we looked broadly in our populations, and we saw that in all the cases, the receptors were completely monomorphic, and they were subject to gene-specific sweeps on the population level. So contrary to this expectation that you might have that a virus would induce diversifying selection on the receptors, they were completely the same. So there's really a tug of war going on, and I think that the selection by viruses is actually not that strong on average in the environment, probably, because otherwise you would probably see some more diversity on the receptor level. And what I'm showing you here is these protein coding genes, and here also these lipopolysaccharides. Here they're not as monomorphic, but you get the same lipopolysaccharide coding on the population level, and meaning that all the phages can attach to all the hosts and inject their DNA. Now, this matters also for interpreting, I think, recombination amongst viruses, because it's a very common observation that viral genomes are extremely recombinogenic in bacteria. And oftentimes, you can find evidence of homologous recombination. You can find evidence of exchange of entire blocks of genes. Yet, phages are highly specific. So how do those things work together? And I think that the broad receptor diversity explains this very well. What you have is you can have an infection, for example, from a phage that can actually kill the host. And if there's at the same time another phage infecting that is inactivated by a defense system, and that oftentimes happens by cutting the DNA apart, then this inactivated DNA is free to recombine with the phage. And therefore, you get these really incredibly high recombination rates amongst phages. The other thing that this changes a bit the view of our findings in terms of like how we view virus-host interactions in bacteria is that, sorry, I always steal my slides from students, and they always love these animated slides, and I was like, have to push 10 times before the whole picture is there. There's a lot of modeling out there that predicted frequency-dependent interactions between the viruses and the host. And this has resulted in this kill the winner hypothesis. And this goes as follows. So you have a microbial community, and one of the genotypes in that microbial community receives a growth advantage. And if it competes with other organisms, it should or could, in principle, outcompete some of those. So you would reduce the diversity. But because this winner genotype gets attacked by viruses very quickly, and the viruses reproduce very quickly and amplify sort of their own killing signal, the genotype gets depressed. And so overall, it should be a sort of a diversity preserving mechanism. But what we see is actually that the viral resistance is so rapidly changing and has such low linkage across the genome that it's really a game between mobile genetic elements and viruses, we think, and that any sort of properties that might give a particular background genome an advantage over others is unlinked from this viral predation. And so this is a review that Otto Cordero and I wrote some years ago. And it was still at the time when receptor genes, we speculated that receptor genes actually move around very quickly, but we can now change this and say that this is really phage defense islands probably. Okay, so with this, I'm going to finish and just conclude that viruses are incredibly diverse out there, but the interaction matrix, because of this diversity, probably also because of the diversity that we see at the bacterial strain level, is actually very sparse. And that's because the phages are super specific. And the specificity, of course, arises from a combination of really broad receptors and rapidly changing mobile genetic elements that determine this phage specificity. And I think this actually opens the question to me, actually, how do phages really make it out there? Because if the bacteria are so well defended and the interaction matrix is so sparse, then I think viruses actually barely hang on, actually. That would be my conclusion from this. Yet you have this juxtaposition of this incredible predatory activity they're supposed to have. So I think these two don't come together right now, and this is one of the main questions we're currently interested in the lab. Okay, so let me just thank the people whose data I showed you today. The foremost, Catherine Kaufman, who started this cross-infection matrix, Fatima Hussein, Antonio Martin Platero, Javier Dubert, Joseph L. Shabini, and Nina Bartlau, and then some couple of long-standing collaborators. And our funding mostly comes from the Simons Foundation and NSF. And thank you for your attention, and I'll be happy to answer questions if you have any.